cytokines, antibodies, and protein purification tools for your next grant. I'm Amy Mendenhall, and I'm a technical marketing specialist for the catalog products team here at GenScript's. I received my PhD from the University of Michigan, where I studied neuroblastoma cancer using cell culture and molecular biology tools. Today's webinar will show you how to incorporate recombinant cytokines, antibodies, and protein purification tools from GenScript into testing a hypothesis that links chemotherapy-related cognitive dysfunction with increased cytokines in the brain, leading to changes in DNA methylation and subsequent alterations in gene expression in the brain. This webinar will highlight methods to test this hypothesis using products available off the shelf from GenScript in animal models. This presentation is designed to show you an example of how you could mix and match products available from GenScript for testing your own hypotheses. If you have questions during this presentation, you can submit them by typing them in the questions field that should be showing on your screen right now. I'll do my best to answer some questions at the end. Questions that are not answered will be posted with their answers on our website, which is www.genscript.com/webinars, along with this archived webinar and links or downloads to any of the associated materials presented today. In addition, you will receive a brief survey immediately after this webinar is over. I ask that you please consider filling it out, as feedback is an invaluable resource for us in improving our services to you. Today's webinar is going to go through a test scenario about writing a grant concerning chemotherapy-related cognitive dysfunction. First, I'm going to give you an overview about this condition, and will present some of the testable hypotheses that could be studied. We will then go through a few suggested experiments using products from GenScript's catalog. Please note that these experiments that we are talking about today are only hypothetical, and we here at GenScript have not tried testing any of these, and we also don't plan to test them or publish anything related to the information in today's talk. Some of the hypotheses presented here today were derived from this review article that was published in Cytokine. It came out earlier this year, and the ideas were presented by Wang's group from the University of Hong Kong. The full article and abstract are available from PubMed if you're interested in reading it. And throughout the paper, Wang's group refers to chemotherapy-related cognitive dysfunction as chemobrain. So just to not have to keep saying that really long term, I will also refer to this condition as chemobrain. So let's talk a little bit more about what chemobrain is. Chemobrain is the subjective experience of cognitive deficit at any point during or following chemotherapy. Patients complain and show impaired daily function in attention, concentration, forgetfulness, word finding, multitasking, and organization. Chemobrain onset occurs during or following chemotherapy and is not due to having cancer itself. Sometimes the symptoms may even last a while after chemotherapy has ceased. And there seems to be a relationship to other somatoform symptoms like anxiety, depression, fatigue, and overall health-related decline. This means that patients may also present with these other symptoms in addition to reporting the cognitive deficit. Interestingly, there is a discordance between the subjective experience of cognitive dysfunction and the objective neuropsychiatric measurements. For example, patients who report symptoms of chemobrain don't necessarily perform poorly on neuropsychiatric evaluations. The cognitive impairment in chemobrain is very modest, and it is also therefore difficult to measure accurately and difficult to study. Traditionally, there have been three main tests that are used to evaluate patients for chemobrain. The Hopkins Verbal Learning Test Revised offers a brief assessment of verbal learning and memory including recognition and recall. The test involves presenting the subject verbally with a list of 12 words. The subject then recites as many of the words as they can remember. This is done 3 times in a row using the same list of words. And then, after a 25-minute break, the subject is asked to recall as many of the words that were on the list. Lastly, the subject is presented with a list of words individually, and after each word, they are to indicate yes or no if that word was on the original list of 12 words. This test basically helps to identify dementia in patients. The trail-making test is a neuropsychological test of visual attention and task switching. It consists of 2 parts in which the subject is instructed to connect a set of 25 dots as fast as possible while still maintaining accuracy. It is basically kind of like a dot-to-dot puzzle that is timed. It can provide information about visual search speed, scanning, speed of processing, mental flexibility, as well as executive functioning. It is also sensitive to detecting several cognitive impairments, such as Alzheimer's disease and dementia. The controlled oral word association of the multilingual aphasia examination, also called the COWAT, is a measure of verbal fluency. The COWAT uses the 3-letter set of C, F, and L to assess phonemic fluency. Individuals are given 1 minute to name as many words as possible beginning with one of the letters, and then the procedure is repeated for the remaining 2 letters. The total number of words across all 3 trials is considered to be the score. Collectively, these tests measure learning and memory, processing speed, and executive function, which are routinely used to evaluate Alzheimer's disease and dementia. However, Alzheimer's disease and dementia may present differently than chemobrain, and so it's unclear whether these tests are the most appropriate tests to measure chemobrain. The current hypothesis of chemobrain is that it's a result of neuronal injury with consequent inadequate repair, abnormal brain remodeling, and also corresponding neuroendocrine immunological changes. However, there really is a lack of supporting evidence in support of the current hypothesis. First of all, neuroimaging studies that have looked at structural and functional changes in the frontal cortex and the related white matter tracts are implicated in executive memory and function. However, the methodology and the small sample sizes of these particular studies don't demonstrate causality for chemobrain, and they also don't really show neuronal injury. Additionally, a direct causality and neuronal injury are not answered by the scientific literature to date. Data on oxidative stress, neuronal repair, immunologic or endocrine changes are not currently available in the literature. So there is a new emerging hypothesis about what causes chemobrain. And this new hypothesis is that epigenetic changes are induced by cytokines, and this can cause chemobrain. The hypothesis is that chemotherapy causes a shift in the cytokines in the body, which then cause epigenetic changes that can persist after chemotherapy has ceased. The epigenetic changes may alter neuroplasticity and cause the subjective experience of chemobrain. So why do we think that cytokines are to blame for chemobrain? Well, first of all, cancer patients who have received immunotherapy using interleukin-2 or interferon alpha have reported an experience of dyscognition, depression, and fatigue. And additionally, the literature shows evidence of a correlation between altered cytokine levels and the experience of dyscognition. This table was derived from the long publication that I mentioned at the beginning of the talk. And as you see here, patients treated with various chemotherapeutic agents are shown to have changes in their different cytokine levels. And different studies have looked at different cytokine changes. So the references are all shown here. And at the end of this presentation, you can see a list of all of the references we used in this particular presentation. So here, some people have seen increases in TNF-alpha, increases in interleukin-6, and there's another study here showing increases in IL-6 and IL-8. All of these different studies show cognition changes, so things like reduced memory performance, memory complaints, patients complaining of feeling heavy-headed and muddled thought and having forgetfulness. So why do we think that epigenetic changes could be the mechanism behind chemobrain? Well, first of all, the cytokine levels are short-lived. So when they change, it's just a short change. But chemobrain often lingers. The literature has shown that DNA methylation and histone acetylation are involved in brain development, memory formation, and the regulation of learning and memory. Thirdly, learning and memory impairments in an animal model have been associated with histone acetylation and also decreased histone deacetylase activity. And lastly, there has been an association of chemotherapy-induced neuropsychological disorders and epigenetic changes in breast cancer patients. So what can we do to test this new hypothesis in the lab? Are there ways to make testing more standardized and controlled? So the new hypothesis, again, which I went over before, is that chemobrain is the result of epigenetic changes that persist caused by a shift in cytokines related to chemotherapy. These epigenetic changes alter neuroplasticity, thus causing the subjective experience of chemobrain. So that's what we are going to try to test in this pretend grant. So one way to make our study more controlled is to move into an animal model. This would allow for controlled methodology and adequate sample size. The behavioral analysis can be studied using standardized learning and memory tests. The biochemical assays can be studied by analyzing tissue samples, and anatomical and histological studies can easily be performed. Since our studies may require some dissection of specific brain regions, which can be very small, we can assume that we could use rats, which are slightly bigger than using mice, and they're easy to get. So here are some questions and specific aims that we would like to look at in this particular grant. So first of all, are cytokine levels changed in the brain after treatment with chemotherapeutic agents? The specific aim would be that cytokine levels are altered in rat brain tissue after exposure to chemotherapeutic agents. A second question that we have is that when rats are exposed to chemotherapeutic agents, how do they perform in behavioral tests that relate to learning and memory? And does direct administration of specific cytokines into the rat brain also cause a similar behavioral pattern? So here we will expose rats to either chemotherapeutic agents or specific cytokines, and then look at negative changes in behavioral tests and compare these to control animals. Lastly, what epigenetic changes are found in the brain of chemotherapy-treated or cytokine-treated rats? And here we're going to look at increased DNA methylation in rats that are treated either with chemotherapy or cytokines or control. So let's look at how some of GenScript's products can help us test these hypotheses using some suggested experiments. So let's look at specific aim 1 first. So here we're going to look at cytokine levels in rat brain after exposure to chemotherapeutic agents. So we're going to have a few different groups of animals here. The idea behind using a rat model of breast cancer, which I'm going to talk about in just a second, is that it isn't the cancer alone that causes the epigenetic changes. We want to make sure that it's the actual chemotherapeutic agent that's causing these problems. So healthy rats, these are just regular rats. We're going to treat them with 2 different chemotherapeutic agents, One is doxorubicin plus cyclophosphamide. We're going to call this AC. And the other treatment will be cyclophosphamide, methotrexate, and fluorouracil, which is called CMS. And we'll also have some vehicle-treated rats to use as controls. The transgenic rats that have induced breast cancer, we're going to treat them with the same treatment and also controls. So I'm going to be referring to these as healthy rats, and then transgenic rats with induced breast cancer, I'm going to refer to those as sick rats. So what kinds of changes are we expecting to see? Well, from the literature, we've seen that the AC chemotherapeutic agent causes increases in interleukin-6 and interleukin-8. And the CMF treatment causes decreases in IL-6 and IL-8. So here, we are going to assay the levels of interleukin-8, both in serum and in homogenized brain tissue in the different treatments of rats that we described before. We will use do a time course using ELISA with an anti-IL-8 antibody. Also, we'll do an SDS page gel using GenScript's Express Plus page gel. And we'll follow up using Western blots with the anti-IL-8 antibody for quantification. And any products that we're using from the GenScript's catalog are highlighted here in green. When I'm done talking about the science, I'll go through where you can find these various products. So here, this is just a screenshot showing the IL-8 antibody. You can get it from mouse. It's monoclonal, and you can read the information on GenScript's website. So there are a couple of limitations here. If IL-8 is only changed in specific brain regions, it might be necessary to pool those brain regions from rats to measure it. And we might also want to check the literature to see where exactly IL-8 is expected to be expressed in the brain. But what would we expect to see in this particular experiment? Well, first of all, we know that the AC treatment has been shown to increase IL-8. So when we look in rat brain and in serum, we're going to expect to see increased IL-8, both in the healthy and the sick rats. And then the CMF treatment is supposed to decrease IL-8. So we should be able to see decreases in IL-8, both in rat brain and in serum, and the same changes in the healthy versus the sick rats. This is an example Western blot using GenScript's anti-IL-8 antibody. IL-8 migrates at 8.5 kD, and you can notice how clean this background is, and you can have a nice titration here of IL-8. So even at low amounts of IL-8, you can still detect it very nicely on a Western blot. Let's move on to specificity number 2, which is that exposure to either chemotherapeutic agents or specific cytokines cause negative changes in the behavioral tests compared to controls. In this case, we need to have a learning and memory task that is meant for rats. And the most commonly used memory task for rats is the Morris water maze. What this test is it's a large pool, about 4 feet in diameter. You fill this with water and also put dry milk in there so the water turns white, makes the water opaque. And inside of this pool is a clear platform. And you put the platform in a known area, although this known area can change during different experiments. And then you place the rodent in the water, and the animal has to basically find the submerged platform. The test assesses the length of time and the spatial pattern to find the submerged platform. So we're going to use this Morris water maze in our experimental groups. This time, we can treat the healthy and sick rats with the same treatments as before, so the AC and the CMS. We're also going to treat them intracranially with TNF-alpha, recombinant TNF-alpha that's made from GenScript's catalog. And we'll also have some vehicle-treated rats. So one of GenScript's main product lines is recombinant proteins, including recombinant cytokines, and I'll show you how to get to those on our website as well. In this case, we would expect that animals that have received either the chemotherapeutic agent or the TNF-alpha would perform more poorly than animals that are vehicle treated. We also would expect that healthy and sick rats should perform approximately the same way, depending on their treatment group, because the cancer rat should not be having the chemo brain. So let's now move on to specific aim number 3. In this case, we want to look at DNA methylation levels in rats that are treated either with chemotherapy or with cytokines. In this case, after treating the rats, the same groups of rats, with the chemotherapeutic agent or the TNF-alpha or the vehicle, we would dissect the brains or separate brain regions from treated animals and homogenize the tissue. We'll then incubate this with an antibody to methylated cytosine. So this is going to get methylated DNA. And then we will immunoprecipitate this using protein A resin from GenScript's catalog. The protein A resin binds to the antibodies and pulls it down. From this result, we will do next-generation sequencing on the DNA to identify the methylated genes. So let's talk a little bit more about GenScript's products here that we've used in this particular pretend grant. First of all, GenScript has a lot of primary antibodies. They have a very high affinity and a very high specificity. They include both monoclonal and polyclonal antibodies, and they have over 1,000 available. Most of these have been validated for multiple applications, including ELISAs, Western blots, and immunoprecipitation. Also in this grant, we were using the Express Plus page gels that are available from GenScript. And these are compatible with most gel tanks. It saves you a lot of time, so you don't have to spend time pouring gels and making the reagents to pour your gels. You can just take one out of the refrigerator and use it. We have many concentrations available. So we have 3 different types of gradient gels and 3 different types of standard gels. And you can see here in this diagram how the proteins are expected to migrate in each of these types of gels. The Express Plus page gels come in 20 per package, and they even come with their own MOPS running buffer. They have a very long shelf life, up to 1 year, as long as they're stored properly. And the pricing is very low. They're only $4 a gel. So this, in addition to saving you time, it's also saving you a lot of money. And if you would like to try this, we offer free samples of the Express Plus page gels. GenScript also has a lot of recombinant proteins, including recombinant cytokines and growth factors. We have some of these produced in E. coli, others are produced in eukaryotic expression systems, and all of them are experimentally validated for activity. They have low endotoxin levels. They're provided lyophilized, which makes for easy shipping and easy storage. It also enables you to reconstitute them in a concentration that is most appropriate for your particular assay. Again, they have a long shelf life, and we can provide them small scale to you or on an industrial scale if you have larger experiments. And the species-specific homologs are often available. We often carry human, mouse, rat, and even many other species as well. GenScript also provides a wide variety of protein purification resins. The one I talked about today was protein A, but we also have protein G and L varieties. These are excellent for affinity chromatography, for protein, or for antibody purification. It's a fast and simple procedure for either batch or gravity flow operation. They have a high binding capacity with a low, ultra-low nonspecific binding, and they're compatible with many different buffer formats, sample formats. And it's also very cost-effective because you can reuse the resin several times. Some of the follow-up studies that we could do on this particular pretend grant would be that after identifying the methylated genes, we could use cell culture models to further study molecular pathways that cause methylation. And different panels of antibodies to cytokines could be designed for ELISA studies and screening assays. So let me just show you where you can find GenScript's products. So if you go to the GenScript website and you click here on the products header, these are all of the products that are available from GenScript's catalog. So all you have to do is search for one of these, click on it, so one of these cytokines, and you can see a full listing of all of the different cytokines that we offer. Same thing for primary antibodies and protein purification tools. So I'm now going to open the questions box and start answering some of your questions. Okay, so if there are any questions, I can answer them now. And if there are no questions, then and you think of one later, you can always send us an email or give us a phone call. And we can answer your question that way as well. Okay, so I don't see any questions. So I'm going to end today's webinar. I wanted to thank everybody for coming, and hope you have a nice day. Bye.